start off kind of where we left off. Talking about T-cell development, T-cell maturation, and the thymus. And a couple people had questions about that, and so I put this figure into the study aids section. This figure is another figure, and kind of really, breaks it down. And so this will simplify things, make it a little bit more understandable. So you have T cells, which haven't even decided whether they're going to be CD4 or CD8s. They're not cytotoxic or helper T cells. They haven't differentiated to the point where they have a specific T cell receptor. They're these T cell precursors, and they will basically traffic to the thymus following chemokine signals and traffic to the thymus, enter the thymus, and then they will become these, they start off double negative. Through certain signals, they basically turn on and make both the CD4, so they become double positive. They become CD4s and CD8s. And then in this cortex, the outer portion of the thymus, they're going to come into contact with antigen-presenting cells, these cortex thymic epithelial cells.
What's a TEC?
So cortex, cortical, so it's in the cortex, thymic epithelial cells. So specialized epithelial cells that are also going to present antigens. And so they're going to express both MHC class 1 and MHC class 2 molecules, which are going to, again, these molecules hold antigens up for presentation. And because they're double positive, they have both, and this is where positive. So the events that happen is first you have positive selection happening in the cortex, double positive, and it kind of makes sense because positive selection is when you have both CD4 and CD8, and so they're going to kind of be perusing these MHC molecules with antigens, kind of just looking to see which ones actually have some specificity. So a lot of, and even in here, and so it is a little bit muddy because if they bind too strongly, they are deleted in this portion. If they bind, so it's kind of the Goldilocks zone. They might bind just right, then they'll move on. They'll become single positive cells. So if you've got CD4, so on a helper T cell, and it finds this MHC class 2 molecule and it happens to recognize it or perhaps some affinity for it, some binding strength for it, then it will move on. It will become a CD4 T cell. It'll stop expressing CD8. It'll make that kind of critical decision. And now it's going to go through negative selection where now if it sees antigen and it binds very strongly, it's going to be deleted. So, and then once it has become got through this selection process, now it goes off into the periphery. It's going to circulate through the blood, through the lymph, back through the blood, lymph, and just do that circuit until it finds its cognate antigen or until it dies. Any questions on that?
So,
yep.
You said when they receive a cytokine, they move to the thymus, right? All these double negative cells move to the thymus. So then they do the positive selection. So if they are double positive, they are strongly binder. So then they delay the kill. So how about the double negative? Do they survive the cycle, come back and recycle, or they dead?
Double negative cells?
Yes. Yeah.
Those double negative cells are always going to go through and become double positive cells in this situation.
What's their lifespan for a double negative?
It's going to be very short, and they're just going to make, so a double negative, you're finding a double negative in the periphery is really rare.
So that means they die in a cortex.
Well, they they either go through this process or they die, pretty much, yeah.
So I heard somewhere said 95% of the T cells die. You know,
Yeah.
only a very few percentage.
The majority of them do.
Yeah, so that's definitely die in the cortex.
Yeah. Well, they're going to die,
Okay.
but they're going to become double positives before that happens.
Oh,
So the double negative is just a stage. It's just a step in that progression. They're going to go there. They start out double negative, then they upregulate their T cell receptors. We're going to talk about how do they make that specific T cell receptor. So there's certain events that are happening in the germline, in the chromosomes themselves. Some rearrangements happening. And we're going to talk about those rearrangements that are happening in the further chapters. But for the most part, all you need to know is that once they develop these T cell receptors, each T cell makes one T cell receptor, and it's fairly unique to that T cell. And there's a huge array of different T cell receptors that are possible. So. But yeah, T cells are going to go through this process, and they're either going to die, they're going to come out and hopefully they're going to have a unique T cell receptor that doesn't recognize self-antigens, or they're going to, in the rare occasion, they're going to come out and maybe have specificity to self, but that's a very small percentage.
so this is the selection double positive is for self and right.
Well, it's non-specific. It's what we're selecting against is the positive selection is selecting for some binding or some recognition of MHC molecules. And the negative selection is selecting against recognition of self-antigens, proteins that are made by our own cells, peptides that are representing who we are, genetically speaking. So, these specialized cells, these MTECs, which are just medular thymic epithelial cells, are going to present a really huge array of, they're going to be making proteins, all the proteins. These are their job is to present everything. So now those are the primary lymphoid organs. These are where we're developing. So remember the bone marrow was where B cells developed. Thymus is where T cells developed. And now they've gone out into the periphery. Now they're looking for antigens in the secondary lymphoid organs. First and foremost of which is the lymph nodes. And so as you can see, we have our lymphatic system is really, so if you take an AMP or AMP2, you know that our circulatory system basically provides nutrients to every cell in the body. Those lymphatics are pretty much entwined with those capillaries of the blood, and so you have basically the lymphatic system that follows along that same line. It's going to be throughout the body, so that if we have an infection, if we get poked by a nail, it doesn't matter where it happens, we're going to have capillaries and lymphatics associated with that. The lymphatics now are going to be this part of this secondary or lymphatic system or immune system, and they'll be delivering antigens to lymph nodes. And so the lymph nodes, so again, our lymphatics are going to drain to lymph nodes, and lymph nodes, so our secondary lymphoid organs are going to be these lymph nodes. The spleen, used to be believed that the spleen wasn't part of this, but now it's been included. And I'm not sure at what point they started including it, but it was within the last few years. T cells and B cells are going to circulate to through these lymphoid organs. They're going to be looking for antigen, and They are going to be separated, once they get into these organs, they're separated into distinct microenvironments. So you have B cell rich areas, T cell rich areas, and they kind of have different roles and what's going to happen there. And the cells migrate towards each other once they're activated. So one thing you have to remember too with B cells is for a B cell to become activated, not only does it have to recognize its antigen or its target, but it also needs to receive help from our helper T cells. So it needs two signals. Once it receives those two signals, then it will become active. And so they have to be able to interact with each other, which is kind of a fail-safe. You don't want B cells just becoming activated. You want to be sure when you activate a B cell, you don't want it to be, well, maybe this is self-antigen, maybe this isn't. We really want to be able to activate it with some context. Okay. Differentiation into effector cells takes place in the follicles and in secondary lymphoid organs, T lymphocytes. So your CD4s, your helper T cells, and your CD8, your cytotoxic T cells, are going to become stimulated, become effector cells or memory cells here in these places. So lymph nodes and spleens are pretty highly organized. They have, again, let's see, I'm going through this again. Let's skip ahead. Looks like I'm on a loop. Must have accidentally copied this. So now here we have, basically, we break down the lymph node. So here's our lymph node. We have our efferent and afferent. So we have our entry. So our incoming afferent lymphatics are coming in, and this is where we have dendritic cells, macrophages, and antigen coming in from the lymph nodes. And as we saw, the lymphatics are all throughout the tissue. So again, if I have a nail wound, I'm going to, antigens or foreign antigens, bacteria are going to enter into that wound. I have lymphatics that are being drained. So I have fluid that's draining into the lymphatics that's going to carry anything from that wound into the lymphatics. It's going to carry it into my lymph node through this afferent lymphatic. And then we have our outer cortex. Okay, so this is the outer cortex is going to be rich in B cells, dendritic cells. So these are the antigen presenting cells. These are what are going to pick up that antigen. So we have antigen coming in. We have some dendritic cells that might be bound to that antigen and kind of holding it and bringing it to this area where now it's going to either, that antigen can either activate B cells, it can bind to the antibodies that are the receptors on B cells, or it can be phagocytosed and then processed and chewed up into little pieces and presented via the MHC molecules on the antigen-presenting cells like dendritic cells or even B cells. So B cells can themselves have their receptors stimulated, but they can also phagocytose that antigen or that chunk of protein or whatever that happens to be, process it and present it to T cells. So here's where the kind of the little dance between B cells and T cells happens.
Where are the T cells?
So T cells are going to be in this. No, they're going to be in the paracortex or the middle kind of section. So that's the next paracortex. And here it says T cell zone. So the B cells that bind to antigen, they're going to get that primary receptor, but they're going to and we're going to talk about this in a little bit. That B cell receptor, once it binds to its target or its cognate protein or antigen, it's going to create a signaling. It's going to signal to the cell. The cell is going to receive this first signal, and then it's going to migrate to the T cell zone. So it's going to migrate to the T cell zone, and in that T cell zone, the B cell is going to now be, instead of being a cell that's looking for a signal, it's going to be presenting. It's going to phagocytose. So there's going to a signal that basically internalizes those receptors. So the receptors get brought into the cell. Whatever is bound to those receptors gets brought into the cell. It gets chewed up. it gets presented via MHC molecules to now T cells. And so if a CD4 T cell now recognizes that antigen, it's going to activate that B cell and give it a second signal. And once that B cell receives that second signal, it will start to proliferate. It'll start to, some of them will become plasma cells. Plasma cells are just a fancy name for B cells that are kicking out a lot of antibodies. And so then, those plasma cells will move to the medulla. And here you see the medulla has this efferent or outgoing lymphatics. So they'll start pumping out antibodies out into the periphery through this efferent lymphatic system. Some B cells will move back. Some B cells will move back to the cortex and into these follicles. And so we have follicles that once they get B cells into them, they make this germinal center here we have. And if we have a follicle here, we have a germinal center, and then inside that germinal center are going to be B cells that have been stimulated that are dividing. And so you have what's called a secondary germinal center would be one that's active with B cells dividing in it. And a primary germinal center would be one that is, for example, we have some germinal centers that don't have active proliferation, and so they would be a primary germinal center. Something to keep in mind. So, you have a lot of these follicles formed, and like this would be a secondary. So the really dark ones that are really well formed would be a secondary germinal center, and a primary germinal center would be where you don't see, you have some, it's a follicle, you have follicles, they're just not well defined. And yeah.
Does each specific germinal center only produce one type of antibody?
That's a good question. I think that, I don't know that they have one, but that maybe they might have a very narrow range of them that are responding to an infection. Depending on how broad the infection is. So some infections, there's one antigen that's like just going gangbusters, and so yes, it would be pretty monoclonal. But there are other infections where there's multiple antigens within that bug that do a really good job at activating B cells and T cells. And in that case, you probably have as well those other clones, but it'd be like you'd have 4 or 5 versus.
So they become active? Like, will they switch from primary to, I don't know if I have to go with
the follicle?
Yeah. Will it switch between being primary and well-defined? Based on
yeah, so these follicles will form. You get all these B cells that are in there, and this takes 4 to 7 days after you have an infection. And that secondary follicle will form. There's full of B cells that are dividing, and they'll keep dividing for about 3 weeks, and then after that, they'll go their merry way. So, they're going to go out either into the periphery. They're going to continue to populate maybe that lymph node with memory cells. So the memory cells that stick around in the lymph node, those are going to be your kind of your primary effector cells, and effector cells that go off into the periphery and are going to be under the skin, or they're going to go to the location of the infection, which happens a lot. They'll just kind of hang out there looking for antigen, and those are going to be your effector memory cells. But yeah, it kind of is a very dynamic and coming and going.
So T effector, where do they go?
Oh, we're going to cover that next. So the T cells. So T cells are not coming in through our lymphatics, our afferent lymphatics. T cells are coming in through the bloodstream. They're coming in, and as they come into capillaries that are going to populate these lymph nodes, they are going to enter through these HEVs, or high endothelial venules. So these are venules that have these specialized, they call them high endothelial because they're a little taller than normal endothelial cells. They allow for the T cell, and so you have, they secrete chemokines that basically tell T cells, hey, you want to hang out. It's like a rest stop. So, hey, there's a rest stop ahead, and then they pull off the freeway into the rest stop, which basically they're going to come into this lymph node. And then they're going to get into that paracortex, and there are this conduit system. FRC is the follicular reticular cell conduit system. So these are cells that basically form this maze, you might say, or conduits, where basically you have dendritic cells that are going to just kind of latch on and surround them, and the dendritic cells are our antigen-presenting cells, and they're going to present antigen. So as the antigen's being brought into the lymph node, it's being presented, and then the T cells are coming in through the blood. They're coming in through these HEBs, these high endothelial venules, and then they are coming in. They're coming into this paracortex region. So they're going to traffic to that paracortex region where they are going to now basically roll and move along this maze and hang out for up to 24 hours just looking at antigen. So they pulled off on this rest stop. It's really interesting, and they just want to see everything. So our rest stops aren't usually interesting, but if they were, this is, we would hang out there a little longer. So
what's the FRC?
This is the follicular reticular cell conduit. So these cells make follicles. They're reticular cells in origin, and they basically make these long fibrous pathways. And so it's more of their role isn't as antigen-presenting cells or anything like that. They're just a conduit system to make a scaffolding by which T cells will just roll along, looking at the APCs, looking at the dendritic cells in here. So here's your dendritic cell, and it's going to present whatever antigen it's come across, and your T cells are going to just keep rolling around until they find antigen. If they don't find antigen, they're going to move to the next lymph node. And here we see we have a similar network that's in that cortex. So within the cortex, the outer cortex itself, we also have these follicular dendritic cells, so FDCs. So these are actually dendritic cells that are now going to kind of create a matrix where B cells are able to kind of populate, spread out, and also look for antigen. So they're hanging out waiting for antigen. T cells are rolling around looking for antigens being presented to them. And so you have this kind of system that's really set up with microzones that each have a function. The microzone here is where B cells are hanging out looking for antigen. Dendritic cells can process antigen, or they can present antigen to the B cells. So it's just a place where basically you're processing what is being made in that region, and it's very local, which is important because, for example, there was a group that was making cancer vaccines, and they were making T cell vaccines, and these T cells were being grown outside of the patients, and they were being expanded and made specific for cancer cells, and then given back to the patient. And then they found that if they gave them back to the patient just in a convenient vein, then they had this really mediocre response. But if they gave them back to the patient locally, where the tumor was, if they had head and neck cancer, if they gave them to where that vein was going to feed into the lymphatics that was closest to that infection or that cancer, then they got really good responses. So not only are the lymph nodes looking locally, but they also can produce much stronger responses locally. So those T cells that then find their specific antigen will, and if they find it in the right context, will become, they'll start to divide. They'll become either effector cells that are going to attack something, or they're going to become a memory cell, and they're become a helper cell, and they'll further stimulate B cells to start producing antibodies, and then those T cells are going to go, then they're going to hang out. If they find antigen, they'll hang out for days and just divide, and then go off, and those effector cells will go out into the periphery and follow chemokine signals and go to the site of inflammation of the infection and target those cells. So, any questions on lymph nodes? So spleen, very similar organ. One of the major differences between the spleen and the lymph node is spleen is much larger. The spleen also doesn't have lymphatics feeding into it, and therefore people used to think, well, the spleen, when they were looking at the spleen, it had lots of red blood cells in it, had some white blood cells in it, lots of platelets in it, and the cells that produce platelets, and they thought it was just this reservoir. It turns out that it's more than a reservoir, and it's basically responsible for blood-borne pathogens. So antigens that get in the blood are going to now circulate through here, or viruses that infect blood cells, or bacteria, maybe even parasites that infect blood cells. So there are certain things that target blood cells or get into our bloodstream, and the spleen is the prime or the secondary organ responsible for trying to get rid of those infections, or at least alert the immune system to it. So, and to do that, it's, again, we don't have lymphatics, but we have these red pulp and white pulp regions. Obviously, the red pulp is going to be full of lots of red blood cells. These red blood cells, if they were infected, we're going to circulate. Red blood cells are constantly being circulated through the system, being killed, replaced. So these red blood cells are getting phagocytosed. They're breaking down, and they're releasing whatever was in them. And if they were infected with something, they're going to release that parasite or whatever it was. And so they're delivering their antigen load to the spleen. And they are going to be in these red zones, and then the white pulp here, or here you can see, is going to be our lymphocyte-rich zone, broken down into this these two different zones. So, for example, we have our PALS region, and we're going to have our marginal zone. So the marginal zone is where your B cells, again, the outer cortex of this is going to be filled with your B cells, dendritic cells, macrophages, and then your PALS, which is perioarterial lymphoid sheath, but that's basically going to be filled with your T cells, and so B cells that become activated are going to move into that T cell rich zone. And you also have follicles that form, and those follicles will have B cells that will migrate to the follicles if they become activated. Interesting thing, before they knew that the spleen was involved in blood-borne pathogen removal, I knew somebody who had a condition called ITP, immunothrombocytopenia, which is basically an autoimmune disease where your immune system's attacking your platelets. And so you're basically removing all the platelets, and you're getting bruising. You're going to have the potential to bleed out if you ever have a scrape. And so in really extreme cases of ITP, they used to remove the spleen, and it would work, and they would get better. And their initial reasoning for removing the spleen is they thought it was a reservoir for thrombocytes which produce platelets. And so they thought, well, if we get rid of the reservoir, we'll increase the number of platelets that are circulating. We won't be storing those platelets we want. I always thought that was kind of a silly thing, because usually your body, if it needs something, is going to release those things. But I don't know if there's been a study on it, but it seems like probably what's happening there is where you're getting B cells and T cells that are removing thrombocytes, and where that's happening is in the spleen. So you're having this response that's happening in the spleen. You remove the spleen, you no longer are having that response. And so chances are that removing the spleen was actually removing the cells that were inducing that autoimmune disease. And with that knowledge, we could say, well, let's not remove the spleen because the spleen might be important. Let's go ahead and try and maybe suppress the immune response that's happening in the spleen at the time and see if we can deal with it in that way. But again, people thought the spleen was important because adults would get into car accidents, they'd get their spleens removed, and they'd be perfectly healthy. The problem is most of the times, if you remove the spleen earlier on in a child, for example, then that child has an increased risk for bloodborne illnesses. Any questions on spleen? Another really important secondary organ is going to be the mucosa-associated lymphoid tissue that lines our mucous membranes. That could be the nasal, that could be lung, that could be our gut. And so the one we're covering in more detail is this gut-associated lymphoid tissue, or also known as GALT. And this is an important layer of defense because we're constantly consuming things or bringing things into our mucosa, and antigens are being brought in. We're eating things that have bacteria in them or other parasites. And so our first line of alerting the immune system that something might be coming into contact with us soon would be in that gut. And it's really great because we usually, when things come into the gut, they're usually going to get killed in that really highly acidic environment, and then it's a great way to deliver antigens to immune cells. So these mucous membranes are lined with lots and lots of lymphocytes. And so here, and anybody who's again taken anatomy and physiology has learned about Peyer's patches that are these kind of special localized areas with lots of lymphocytes, B cells, and T cells. They're just lying in wait, and where they're going to have specialized M cells that create these little pockets. So you've got lots of villi moving things along, absorbing nutrients, but then you have these M cells whose job it is to basically move antigens across them and into the underlying immune cells that are laying in wait, waiting for signals. And the book is going to only really covers and kind of focuses on effector responses. So parasite comes in, it's broken down, the antigen is brought across these M cells and is presented to B cells or these dendritic cells, which are going to then become activated and create a germinal center. But actually, these are probably more often involved in suppressing immune responses to things we're eating, for example. So let's say I really like to eat peanut butter, but I don't want to have an immune response against a peanut protein. So I'm eating peanut butter, the peanut butter comes in, it's getting broken down into antigens, and it's being picked up by these cells. There's no danger because peanut butter doesn't have any innate danger where it's damaging cells. And so I present peanut butter antigens to my B cells, to T cells, and they're recognizing it because I don't have any peanut butter genes in me. So I haven't deleted T cells that are specific for peanut butter. So I see this antigen, and my T cells can decide, hey, this is dangerous. I need to mount a response. The B cells can start producing antibodies. Or I need to decide, hey, you know what? I'm going to be eating peanut butter again next week. Maybe I don't want to mount a response against it. It's not doing anything to me. So most of the time what happens is those cells, as long as there aren't any danger signals being produced, they will actually produce T cells, regulatory T cells that are suppressive, that will suppress potential responses that you might have. And so this is where we started to, and so you have these M cells. Their role is to basically be really leaky and allow lots of antigens to come into these B cells and T cells and do the same thing that happens in the spleen or in the lymph nodes. But it also has the additional role of, as long as there's no danger signals, maybe we're going to induce a suppressive response.
Can I ask you something before you comment?
Yeah.
About M cell? What's their function? They are leaky. So what's
Their function is they're kind of phagocytic, and they move things through the cell into their
inside the cell?
yeah, through basically into the cell, then out of the cell. So it's
Also,
basically
it's trafficking.
yeah. So they're moving antigen, big chunks of things in and out, and that's just they're like little conveyor belts, pretty much.
So they are like a transporter or something? Because it is.
Yeah, and if you look at them, if you were to look at them, they might look like a goblet cell. Those people who, again, studied anatomy, these are specialized cells. Goblet cells are secreting things into the mucosa, but then these cells are basically bringing things.
So they kind of move antigen through the B cell?
Yeah,
So they also move the antibody?
well, so the antibodies are going to be produced on this side.
Yeah.
If there are antibodies in this side, yes, it would move them. So, like if you were to maybe breast milk, let's say a baby is consuming breast milk, those breast milk is chock full of, I forget which IG. They're full of antibodies, and those antibodies are going to be brought in. And brought in. So, yes, they will also bring in antibodies, and then those antibodies can enter that way. But for the most part, they're trafficking antigens. And the antigen, again, is any protein or bacteria or cells or whatever. So let's see. I have time. Yeah, I'm not gonna. So there are some videos that I will put up, and they're really nice, kind of walkthrough, and have some really nice pictures, like this one. I'm not gonna play the whole thing, but I'll just kind of skim through it. And this is put out by Nature Video, so Nature is one of the big journals that puts out really good stuff. And so here's immunology in the gut mucosa. And it's talking about how, kind of, if you go through here, you get these virus patches that are now going to basically be areas that are going to bring antigens in. They have lymph nodes associated with them that are really closely. You have these M cells here. And also what you have are dendritic cells are actually probing, looking for antigens and bringing them down. And usually, and this video really is more focused on that suppressive response. So these dendritic cells, because there's no danger, there's no cell death going on, they will come in and they will present antigen to T cells, and those T cells are going to then become Tregs, so regulatory T cells, not effector cells. They're going to become cells that are going to suppress any other cells that also recognize that antigen. And so those T cells are then going to basically populate areas, secrete suppressive cytokines, and basically create a dampening field to where you're not going to get an immune response. And how do we get around? Well, what if we do want an immune response? So here we have bacteria that are entering, they're killing cells, there's cell damage, there's danger signals. So these Tregs actually will get suppressed depending on the danger signals, and then it allows for mounting an actual effector response. But if that danger signal is not there, for the most part, we're inducing tolerance to a lot of these antigens, because you don't want to have immune responses against cells that you are consuming constantly, or foreign antigens that aren't detrimental. So good video, kind of something to watch later on when you get a chance. Okay, so other secondary, so I guess one other thing to mention about this is another organ that people used to think was not really important was the appendix, and some of you may have asked, some of you asked about that last week. Didn't really want to get into it because we hadn't gotten here. But the appendix, they believe, is a reservoir for good bacteria. And you can remove an appendix. You'll usually be perfectly fine. But they started to find that people that had an appendix were 4 times more likely to, or they were, people who had an appendix removed were 4 times more likely to have a recurrence of Clostridium Difficile, which is a common, what's called a nosocomial infection in hospitals. You go into a hospital and you get put on antibiotics, and then you have this flare-up of this Clostridium, and you have...
So 4 times?
Yeah, you're 4 times
Clostridium infection?
of having a recurring infection. So it's and anybody who knows about Clostridium difficile knows that one of the treatments for it is fecal transplant. So that's one of the grosser areas, but you treat it with good bacteria. So a lot of times you have this flare-up because you have this one bacteria that's part of your normal flora that is resistant to antibiotics. You take antibiotics, it clears out it clears out all the bacteria except for that really bad one that starts to now produce inflammation and really starts attacking things, and you have explosive diarrhea. You can die from it. So what happens is your appendix is kind of this storage of good bacteria that's going to repopulate your gut with bacteria that your body knows isn't dangerous. And how does it know that bacteria isn't dangerous? And how does it put just good bacteria in the appendix? Well, this is how it puts good bacteria in the appendix. Appendix is full of lymphocytes. It's full of lymphocytes that are maybe suppressive towards a response against bacteria that have never done anything to hurt us. So if a bacteria comes in and it does harm, we're going to mount effector cells. And we're going to know it. If we have a bacteria that's in our gut, because our gut is chock full of bacteria, as long as it hasn't done anything to hurt us, we're like, hey, you can hang out, you can come to our bar, you can drink, you can do whatever, just don't get rowdy. If you don't get rowdy, then you can hang out. So they kind of, we see their antigens, we kind of know them, and we produced Tregs, and we populated that appendix with those cells that are basically have never done anything bad to us. So that's how we can do that. Other secondary lymphoid organs, the skin, largest organ in the body, and the skin is going to be, it's going to have lymphatics associated with it very closely. It's going to have blood vessels associated with it. And those who have studied know that there are these specialized Langerhans cells that are within the epidermal layer, and they are just hanging out there waiting for antigens and kind of perusing. Keratinocytes are important because they actually will, upon damage, will start secreting inflammatory cytokines. That's going to bring in white blood cells to kind of look around. It's going to bring in those the antigen-presenting cells, which are then going to present antigens to T cells, B cells that are kind of just hanging out, underlying tissue under the skin. Now, the difference here is the majority of these T cells and B cells are mature. They've encountered antigen before, or they're memory effector cells. In fact, the majority of plasma cells are here. There are more plasma cells in the skin than in all the other organs, even in the bone marrow. So these are where cells that they know who's bad and they're hanging out and they're waiting. Yeah.
When you say APC, you mean
Antigen presenting
Yeah, I
cell.
know.
That can be a B cell, dendritic cell. That can be these Langerhans cells.
oh,
So the Langerhans cells are the resident APCs that hang out and wait. But if there's damage, the keratinocytes they'll send out cytokines and chemokines and basically create this inflammatory environment that's going to cause the blood vessels to become leaky in that area. They're going to, all the white blood cells are going to come in through those blood vessels. If those blood vessels became damaged directly, they'll also allow white blood cells to leak into the area. So other APCs will enter after that stage.
Already cell belong to the dendritic cell, belong to APC.
Langerhans are APCs, and they are a form of dendritic cells. So they're a subset of dendritic cells. There's lots and lots of different types of dendritic cells, and they all have varying types of signals that they produce. So that allows for kind of a more refined responses. Do I want to induce suppression or an effector cell? So that is pretty much that. Let me see if we have time to do this. So, I think what I might do is just try and jump into the next chapter. Any questions on this one?
I have a
Yeah.
few questions. Okay, so whenever you're talking about Peyer's patches, so you gave your example of like peanut protein, would there be like a similar situation for people who have celiac disease with gluten?
Yeah.
Is that
So you're getting it in, the gluten antigens are being presented.
okay?
They're just, for some reason, either and to tell you the truth, I don't know a whole lot of what the theories are behind that, but you're mounting an effector response or an inflammatory response. Probably antibody, probably inflammatory response against that versus suppressive response. And understanding these type of things, that's what researchers are trying to do, is how do we induce a suppressive response? How do we induce tolerance? And most of the time we study how do we normally induce tolerance, and can we kind of replicate that? I mean, that allergy shots are an attempt to do that. So.
And then,
Sorry, I was going to follow up with the second to that question. Is there a difference between the gut associated lymphoid and the mucosal associated lymphoid and allergies?
So, versus all of these are mucosal associated, like gut, nasal. I don't know that there's a difference per se between gut versus nasal versus other mucosal tissues. There is a difference between them and your skin, or them and your lymph nodes. In fact, there are people trying to induce tolerance, and they'll introduce antigens nasally because these malts are associated. I mean, one of their major jobs is to induce tolerance to antigens. So instead of injecting an antigen under the skin, people are delivering it nasally, and in the hopes of delivering it without any danger signals, and trying to induce tolerance in that manner, because that's a natural mechanism by which you can induce tolerance. And so, knowing that, people are trying to induce tolerance by delivering antigens in that area via the mucus. Yeah.
It'll be like the nasal little flu shots that they're starting to give out or whatever.
Yeah.
That'd be an example of that.
Yeah, exactly. And so now we can see that people are taking this basic. So this is why you guys want to have this, so that you understand why would I care about introducing an antigen nasally versus injectable in the back or under the skin in the back. And well, what cells are underlying the skin? Mature effector cells. If they recognize antigen, they're going to probably produce an effector response. And I've just produced some trauma by injecting that. So there could be some variety on, well, this is a very experienced nurse. She has not induced hardly any trauma when she injected versus me. If I were to go, I would probably induce lots of trauma when I was trying to inject antigen. So you probably wouldn't want to get allergy shots from me. But then now, much easier, nasal, taking something nasally, there's not going to be variation in how we deliver it. Unless you are shoving something up your nose really hard, you're probably everybody's going to get it in the same way. So yeah.
Now, whenever you're saying that people who have their appendix have a higher chance of getting recurring infections, because of how most of their micro.
Well, the one we know in particular is CD because they've had studies on that. I don't know how broad that is, but yeah.
Would a way to maybe counter that would be taking that? Would you be able to take probiotics and reduce your? Okay.
So definitely, if you had appendicitis and had it removed, you might be somebody who says, you know what, I don't have this additional protection. I want to maybe take probiotics.
Oh, okay.
So yeah, totally. Yeah. And in fact, doctors hopefully are recommending that nowadays when they do that with this further knowledge. Before they probably wouldn't have. But, yeah.
That's basically what the fecal transplant is, right?
That's what it is. It's
Okay.
here's an appendix, and I'm gonna.
Not as aggressive.
Yeah, it's easier than giving somebody an appendix, but you know. Yeah, that's what it is.
Right now, the clinical for the CD infection is quite important. So they can really, the patient got a CD, it's not dying from CD. It's dying from cancer. The CD can be healed. Can be.
Well, and there are antibiotics that will kill CD. So a lot of times it'll be a combination of them.
Yeah.
So it'll be, okay, we're going to target CD with an antibiotic other than ampicillin-based, and then we'll also give probiotics.
Yeah, usually the patient, they ask to take the yogurt or something like that. Oh, no. Can you do us a favor?
Yes.
Can you United wants to post the lecture PowerPoint online? Like for instance, just talking about Cellular Signal.
So I'll try and get them up on before lecture. I usually get them up by after lecture. So you can go revisit them, but I'll try and get them up before. I might get up. What I get up is usually a base lecture before, but I might add things to it, and then I'll like for example, I added a few slides to this lecture 2, and so I will update it on Canvas today, and so it'll be updated.
I cannot.
And I don't know if it gives you guys notifications when I do put things on.
No, that's okay. I don't see anything about it. Would like to, because I like to write.
Yeah.
It's much easier for us, because I'm not like you, I'm old, I'm not writing. Plus, the computer is nice.
Alright, so here's what I'm going to do. Because we only have maybe 25 minutes or so, I don't know how far into 3 I can get, but we can do an activity to kind of solidify the concepts that we covered. So I might as well. So everybody get your cells. So you're gonna be one group. You 3 are gonna be a group. Let's see. You 3 are gonna be a group here. Can you guys work with? Got a group. You two, and then you in the back. You two, so you guys, you four at the end working that with that one, and you guys work with this one. So pull out your cells and put them out on the table. And we have, you can see you got a TH, you have a TC, you have a couple different B cells, and you have this dendritic cell. You know it's a dendritic cell because of its shape and its dendrites. And then we have these guys, these orange guys. This is our bacteria. And so put on top of your bacteria a bacterial antigen. So that's going to be this shape here. It's going to be our bacterial antigen. And so, yes. So you have bacteria. So let's start with the spleen. Let's start with the spleen. So you have your bacteria. And there's, here, let me. So you've got so we have our spleen, and we have the venule that is going to supply blood to the spleen, and we have our white area here, and it's going to have this outer area and this inner area, and then so out here we have, so our bacteria is going to be brought in. Maybe it gets engulfed by a red blood cell, or maybe it affects a red blood cell. It's brought in, and that antigen is released, and then comes in and binds. So it gets picked up by. So the whole thing, the whole bacteria gets picked up by our dendritic cell.
Oh, where?
So yep. So our dendritic cell is going to pick this guy up.
Where?
So just it has a little binding site. So dendritic cells, they have receptors that pick up things kind of non-specifically. They're really sticky, and they like to bind to antigens, just bind to anything, and they'll bring them in, and they'll present. They'll chew things up, they'll phagocytose it, and then they'll present this antigen to. They're going to present this antigen, or they're going to deliver this antigen to now. They're going to come into this region that's rich in B cells, okay? And that B cell is going to recognize a little chunk of protein that's on that. So the B cell is going to recognize that chunk of protein. So here we want to see that, well, there's lots of different B cells with different B cell receptors. Some of those B cell receptors will bind to this antigen and some won't. And when we're talking about antigen in this case, we're talking about big chunks of something. We're not talking about little peptides or little antigens being presented via MHC class 2. We're talking about maybe something that's on the surface of a cell. So in a lot of cases, these are things that B cell can recognize that's on a cell. So we've got that. So where is this B cell? So this B cell, he sees that signal, and we're going to talk about that in chapter 3, is now he gets tickled on that receptor. How does that receptor now translate that into a response? And so we have a B cell that's receiving its first signal. Where is that B cell going to go now? So that B cell saw antigen in here. So now where is the B cell going to go? Into the paracortex, or if it is a lymph node, or into this T cell rich pallid zone or whatever. So it's going to come here to interact with who?
TH.
Yeah, TH cells or T cells. So then when it moves into this region, it's going to basically internalize. It's going to internalize this, and I don't have an MHC molecule on this, which I really need, because these are now going to become the antigen presenting cell. These are going to actually now internalize that antigen. It gets chewed up through the proteasome, shunted into the endoplasmic reticulum, It's going to get mounted onto an MHC molecule. That MHC molecule is going to traffic up to the cell surface, and now it's going to present antigen and just sit there. And then the T cells, so it's just sitting there, the B cell. T cells are coming around, and they're looking at everybody. And this T cell comes along.
Which T cell?
Well,
PC.
the TH. So TH comes along, and it recognizes this antigen being presented, and it's going to be on MHC class 2. We'll talk about what's MHC class 1 versus 2. MHC class 2 is what's on our antigen presenting cells. MHC class 1 is what's on all the rest of the cells in the body, except for red blood cells and a few like sperm don't have MHC class 1. So a few cells don't have MHC class 1, but for the most part, most cells have MHC class 1. Not all cells have MHC class 2. So not all cells can stimulate these helper cells, but antigen-presenting cells like this B cell can. And so now we've stimulated the TH cell, but this TH cell that becomes stimulated, and we'll talk about that, is going to start secreting cytokines that are going to bind onto the B cell, and they're going to create a secondary signal. That B cell is going to say, hey, not only did I see antigen this first time, but I'm seeing that there's a T cell that also sees this antigen, or at least a piece of it, because usually it's not the same. It's not the same piece. It's usually a smaller little chunk of that, just the tip that might be presented to that. So it doesn't have the same specificity for the same, but it has specificity for something inside that protein. And so now it's sending cytokines. It's secreting cytokines, and it's telling that B cell, hey, I saw it too. So you saw it, I saw it, we both saw it, and maybe you should do something about that. And so the B cell says, okay. And so it starts dividing. Some B cells create germinal centers. Some B cells become plasma cells and start secreting antibodies. And there's that. The other thing that happens is that these antigen-presenting cells are also, so cells that are infected can also stimulate these cytotoxic T cells, and those cytotoxic T cells are going to make effector T cells that are going to attack and kill the target as well. So they're all intertwined. Because we have so much positive and negative selection with these guys are kind of the last, they get the last say as to whether I'm going to mount a response. B cells aren't as selective, so we might have B cells that are specific for self-antigens or things that I don't want to mount an immune response. So if they don't get that second signal, we don't want them to start kicking out antibodies that are specific for our cells. So it's good to kind of review, and have an idea of what's going on, and just have the general overview of that. So we're going to try and cover. So if you could read chapters 3 and 4 for Wednesday, we're going to try and run through receptor signaling and kind of the pathway cascade that goes on with those. And I'm just going to cover the first. So I'm going to try and get us just into what are the important concepts to understand with that. And I'm not going to jump into it a lot. I just want you to be focused on, this is what I want us to know. How do these cells decide what they're going to do? Just based on recognizing an antigen. So you get this cell. First thing I really want people to know is you have a cellular signal. Is basically any event that instructs the cell to change its metabolic or proliferative state. So in this case, T cells, B cells, other immune cells, that even dendritic cells, the antigen-presenting cells. Antigen-presenting cells, they start out their lives collecting antigens But once they get a certain signal, they're going to just sit there and stop collecting antigens. They're just going to present whatever they found. It's like they freeze in time, present all their antigens, and just sit there waiting for T cells. So they're all receiving signals telling them to do something. Do I want my T cell to proliferate? Or do I want it to die? So signals are usually generated by binding of a ligand or antigen to a complementary cell-bound receptor. So that ligand can be an antigen. It can be an antigen being presented via MHC class 2 to a T cell receptor, or MHC class 1 to a T cell receptor. It could be a big chunk of protein bound to that binds to an antibody. It can be a cytokine that binds to a cytokine receptor that is going to tell a cell, like the B cell, that is receiving cytokines from the T cell, that's going to tell him that, yeah, there's not only are you activated, but this T cell is activated too. So that's how they communicate. A cell can become more or less susceptible to actions of a ligand by increasing or decreasing the expression of the receptor for that ligand. T cells have a cytokine receptor for IL-2, and we'll talk about what IL-2 is. But they have a really low affinity receptor for it until they see antigen. And then they upregulate the receptor. So there's no reason to be expending energy making lots of receptors for things unless I know that I need to start worrying about that. So a lot of times cells will upregulate depending on whether they received an initial signal. They'll upregulate a receptor to now look and see, okay, I saw antigen. What is the context with which I see that antigen? Does anybody else see it? Should I worry about this? Are there danger signals? Is there inflammation? So it's not looking for those things necessarily all the time. Ligands may be soluble molecules or a peptide, carbohydrate, sugar, a lipid. So again, antigen is just a word, or ligand is just anything that's going to have recognition on that receptor or different receptors. Ligands can travel long distances from the entry point. So again, you can have antigens in the bloodstream or in the lymphatics that may travel a long way before they reach a cell that is relevant to their receptors. Ligand receptor binding is, so this is all non-covalent. These aren't. And so this is where the term, so look up affinity, what affinity means. So if you know, I mean, any English major or avid reader knows that if you have an affinity for something, you like it, you enjoy it. All this means is basically a receptor that has high affinity is going to bind very strongly to something, and a receptor binding that has low affinity has weak or short, doesn't bind for a very long time. And so this binding is non-covalent, so it's reversible. So it's happening, it's holding on. If it has high affinity, it holds on for a longer time. If it has low affinity, it's holding on for a little bit and then losing that contact. And this is why it's important, like B cells have more than one site. So B cells, they have specific antigen-specific, more than one antigen-specific site. So if one recognizes antigen and loses contact, the other one might bind, and so you increase your chances that it's always going to be bound to that antigen, or that infected cell, or that bacterial cell. Ligand receptor binding induces molecular changes. So once it binds, it's going to induce a change in the receptor. It's going to maybe move that receptor closer to another receptor. And those receptors then will have a covalent change. They'll dimerize. They'll have disulfide bridges that are formed. Or they will change conformation so that they become, and a lot of times what happens is you get conformational changes, dimerization, clustering of planets. So you bring a lot of these receptors together in one location, and they change location in the membrane. There are these portions of the membrane called lipid rafts, and lipid rafts are basically cholesterol-rich areas within the membrane that look like little, and they call them lipid rafts because they're like little rafts that are floating in the fluid mosaic membrane. and they're stiffer, and they're kind of, they make little patches that have different different proteins associated with them than in the rest of the so we can actually create regions within the cell membrane that have molecules that aren't in the rest of the membrane. And if we localize a receptor to those lipid rafts, now those will come in contact with those proteins that are associated with lipid rafts, and we get signaling. So it could be an enzyme that is localized in that lipid raft where it doesn't find its target until that receptor comes in contact with that. So they move locations in the membrane. And that's really important for immune responses. Covalent modifications to the receptor itself. So even though the receptor's binding to the ligand is non-covalent, there can be a covalent modification made to the receptor itself with other receptors or other molecules. Receptor alterations induce cascades of intracellular events. So basically these cascades, these are these huge pathways that have multiple events happening: phosphorylation of molecules, dephosphorylation of other molecules, enzymes that are activated or released from being bound to other molecules, and those enzymes will cause other things like calcium release from the ER. That calcium then binds to another enzyme that then will release a transcription factor. That transcription factor goes into the nucleus and starts making things. And there's multiple transcription factors that enter the nucleus upon activation of a T cell. And that's how T cells can decide, am I seeing antigen in the right context? Maybe they only have one transcription factor that's turned on, it might kill the cell. Maybe they have two that's turned on, it might cause the cell to become a suppressive cell. And then maybe they have 3 or 4 transcription factors that really tell the cell, hey, not only do you want to divide, you want to become an effector cell, you want to do all this. So basically there's different signals, and there's different pathways. Pathways coming from the T cell receptor, pathways coming from cytokine receptors. Lots of different events going on. So it's always good to kind of review those so that you're not seeing them for the first time as I'm popping up the slide next on Wednesday. Cell signaling ends. The end result is often an induction of a change in the transcriptional program, like I said, of the target cells. Sometimes multiple signals. Again, I said that already. Integration of all signals received by a cell occurs at the molecular level inside of a recipient cell. Pretty self-explanatory. We're causing binding of this antigen is causing molecular changes. It's causing phosphorylation events. It's causing transcription factors to enter the cell. It's causing new mRNA to be made. It's causing new proteins to be made. It's all self-explanatory. So next week, we'll kind of start, well, I got 5 more minutes. I can get us through at least 1 or 2 more slides. So basically you have, so how this all works is you have antigen. And this antigen or protein or peptide or lipid or sugar or whatever it is going to bind non-covalently. So here in red we have the carbons, the carbohydrate groups of our antigen receptor or a ligand. That can be a T cell receptor, that can be a receptor for cytokine. And you notice there aren't any lines drawn between the blue and the red, but there are things like, for example, there are hydrogen bonds. So hydrogen bonds aren't as strong as covalent bonds, but if you have lots of them, they can be pretty strong. I mean, you can, water, will it's cohesive. It will, for example, surface tension. I mean, it adds up and can be a pretty strong bond. Ionic bonding. So these are bonds that aren't covalent, but still are, slightly positive portions of this can bind to slightly negative portions of this receptor, and therefore you can have you have ionic bonds that are again weaker than covalent, but when you have lots of them, you can have what's called high affinity for something. Also, you have hydrophobic interactions and van der Waals interactions. These are, again, weaker interactions, but still can add up in other ions. So you have all these that contribute to what's called affinity. And so if your receptor binds to your ligand, and it has a lot of these interactions that really make them stick together for a long time, you have high affinity. And so that is going to basically, the longer two cells can hang out and affect each other, the stronger the signal. And so affinity plays a role in that signaling. The other thing that plays a role is how many receptors and how many ligands do I have interacting. So, and that plays into something called avidity, how avid is something. And so affinity is important, but also avidity is going to be how many receptors do I have and how many ligands do I have? Maybe I have what? Because usually you have one T cell that has one receptor. It's not finding one MHC with one antigen. It has to come in contact with maybe 2 or 3 or 4. I've heard somebody throw out the number, it has to have a minimum of 4 molecules to really stimulate. But there are other molecules that are going to strengthen that synapse between the two cells, that are going to strengthen that binding. And the more of those you have, the higher the avidity is of something. There are some antibodies that have multiple sites, antigen-specific sites. And the more antigen-specific sites you have increased avidity. So you can have a stronger bond to a weaker antigen if you have more binding to it. So there's 2 terms that you need to understand is affinity versus avidity. So affinity is how the specific strength of that binding between the receptor and ligand, and avidity is the multiple effect of having more than one receptor and one ligand. Let's see. So antigen, immune system receptors interactions are enhanced by co-receptor binding. So here, these are separate receptor and ligand interactions that can take place near the original interaction. One example of that is CD4 and CD8. So that example could be, here I have an antigen presenting cell, and it has MHC class 2, and it's presenting an antigen, and the T cell receptor recognizes that antigen. So here's my T cell, and then so here I have my separate receptor ligand interaction can be, for example, CD4. CD4 binds to this non-variant region of the MHC molecule, and it strengthens that bond. So CD4 recognizes MHC class 2. If this were so this is a helper T cell. If this were a cytotoxic T cell, it would be recognizing MHC class 1, and instead of CD4, it would be CD8. And so that strengthens that bond. If we don't have that bond, if I break that bond, then it's going to be a weaker affinity, or avidity is going to be weaker, and I'm not going to have as strong a synapse. These cells aren't going to associate with each other as long, and I could actually suppress the immune response. So if I were to block these T cells with an anti-CD4 antibody, I could inhibit any type of interaction in there as well, and that's actually something to do to suppress immune responses. So let's see. Oftentimes a single type of interaction may be insufficient to lead to an activation event. Again, that depends on the affinity and how many receptors you need. Co-receptor interactions can provide a second signaling interaction. So I'm